My name is Davis Johnson. I'm a sophomore biomedical engineer as well here at Texas A&M. And I had the opportunity to work with Dr. Maggie Chi and some really, really cool people at the MIT Chemical Engineering facility in Boston. So our lab primarily focused on microfluidics. And it is a way of modeling bodily functions in vitro or outside of the body. So a lot of different bodily functions are really, really hard and really expensive to model. And this is a simplified way of seeing that. And these pipelines, so these systems are mini pipelines, 200 micrometers across or 1/5 of a millimeter. And they're able to hold cells and such. And you're able to see how different cells interact. And that's what I studied. So this was my first time lab experience. Oops, sorry. This was my first time ever in a lab, so there was a lot of learning curves and learning basic procedures such as cell culture and learning different morphologies of cells. So throughout my time, I cultured 7 different cells. Um, Here they all are. Most of them are endothelial cells, so they're blood vessel cells, which is really, really cool because they're applicable not only to the retina, but to everywhere else in the body. And the next part that's really fundamental to microfluidics is immunostaining, which tags proteins. So you're able to see which proteins are expressed more. Um, so we had a code that once you stained for these proteins, in this picture, it's occludin, which signifies tight junction proteins, which is fundamental in keeping certain things out of the retina. So if we didn't have those proteins, which can degrade at times, we would see a lot more random, like, those floaters in your eyes, we would see a lot more of those. So being able to model the eye correctly and being able to quantitatively prove that is crucial. And then another area of study is chemical exposure. So another area of research is seeing at what point do these factors called COX-2 and other cytokines is what they're generally known as. At what point do they start affecting cells? So as you can see, CCL3, which is just a protein, really, really affects these endothelial cells at 100 nanograms per milliliter and is hardly significant before that. But these images are able to be quantified using a code that my graduate student made. And then all of this is turned into data eventually. So part of our lab was seeing how much infiltration through microfluidics chips we got by a certain type of cell. In this case, it's T cells, and the more infiltration means the more damage to the retina that would occur in certain situations. So I was in charge of making all of those data sets as well. And here's how we count the data. So we had 24 different images taken, each an hour apart, and we were able to run that through a code and then get percentages of infiltration. My project, though, was called the CoFlow project. It incorporates actually all of these simultaneously. Um, and the goal of CoFlow is to be able to seed multiple cell types in the same channel using different inlets. So this case is just dye. So it's 2 different dyes running through the same chip, running into the same channel. And the really, really cool part about this is since it's on a micrometer scale, laminar flow can be achieved really, really easily. So flows can be separated throughout the channel. It's a lot of fluid dynamics, but it's relatively simple to understand. But we were trying to be cost and eco-friendly. And so we would try to reuse these polymers and such. And so my biggest achievement was actually creating this solution that cleaned out the chip. So anytime that we ran these polymers through here, polymers also solidify in the chip. That was the goal of it. It would totally clog up the chip. and we added a dye so you could see it. So normally, it took 24 hours to partially clean out the chip, and it was really hard to see whether or not it worked. But after I created this solution, um, this is real time. It knocked almost all of it out in 5 seconds. But that was a lot of biochemistry that went into that as well. Um, And then one thing that I found really interesting was there's a lot of parallels to agriculture application. So the CoFlow process aims to be able to put two different cell types together, but letting them interact, but not mixing them. And that's something that's fundamentally found in all life sciences, including agriculture. So one area that I got to explore a lot and got to talk with a professional about was bioprinting meats. And that's one of the main focuses is trying to be able to print two different types of cells at the same time to achieve the same texture of meats. So it can be texturally and like tasting the same as traditional slaughtered meats. But yeah, this was a great preview of a ton of different routes I can take in college. And I learned how to become a collaborative researcher and how a real laboratory works. And then this CoFlow project could implement a different type of solution. Ultimately, we weren't able to get it to work with cells ever. That introduces a lot of factors because the fluid mechanics becomes really complicated whenever you bring into cells because each cell is uniquely shaped and they don't like to uniformly distribute themselves. But it's looking promising. So implementing more cell types into microfluidics chips could be the next step as well, trying out different cells. And then my biggest takeaways was how collaborative MIT was and supportive the culture was. And everyone in the lab group was a great mentor in some way to me. And yeah, thank you for the Science Influencers grant for funding me and the G Lab for hosting me.